Hi, my name is Tejan Kim, and I'm going into my senior year at Hudson High School. This summer, I worked in the translational medicine track of the science internship program in Dr. Qinglong's lab, which is part of the molecular cardiology department. My project was on the case control association analysis of 4 SNPs with atrial fibrillation. Genetic factors contribute significantly to the risk of atrial fibrillation, or AF. To date, there are 17 genetic loci identified for AF by genome-wide association studies, or GWAS. We recently have conducted a GWAS for AF in the Chinese Han population and identified 8 SNPs. In this project, we applied Tachman assay to examine the association of 4 SNPs with AF in the CCF-AF cohort. We found that one SNP, RS2200-733, was statistically significantly associated with AF. The other 3 SNPs failed in replication, which might be due to relatively small effects in the CCF-AF cohorts. In the future, larger samples are needed to confirm such associations. Atrial fibrillation, or AF, is the most common form of cardiac arrhythmia, characterized by a very rapid, irregular heart rate. Familiar AF is a significant portion of all AF cases, with heritability of AF estimated at up to 62%. This high incidence of familiar AF suggests that a genetic variant is a major risk factor of the disease. To study such a possibility, GWAS, or genome-wide association studies, are used. GWAS is a systematic and unbiased method to detect genetic variants associated with complex diseases such as AF. We have recently conducted one GWAS for AF in the Chinese Han population and identified 8 significant SNPs. Replication of the above 8 SNPs in other populations, such as European populations, is important to finding whether the effect of these SNPs are concurrent in other populations. So SNPs, or single nucleotide polymorphisms, are variations in single nucleotides occurring at specific positions within the genome. These common variations cause each individual to be unique in appearance and function, and also cause the similarities seen within families and close relatives. So if you look at the image, you can see that there's 2 DNA strands pictured, and they are exactly the same except in the rectangular box, where the top one shows an A and the bottom one shows a G, and that is the single nucleotide polymorphism. There are 3 ways to genotype SNPs. HRM, or high resolution melt, is used for relatively small sample sizes. The cheap cost and accuracy of HRM makes it an effective genotyping method. However, the RT-PCR machine used for HRM was having problems, so we used Tachman gene expression assay instead. Tachman is used for larger sample sizes. It's more expensive than HRM, but it's slightly more reliable. This is the method we use as it met both our budget and sample size. Microarrays are useful for analyzing millions of samples quickly and are used for GWAS studies. These microarrays have been used in GWAS for AF. In 2007, the first genetic locus was identified for AF in the location 4Q25. Since then, 17 genetic loci have been identified by GWAS. However, to gain a more complete understanding of AF, more SNPs and their functions must be identified. In order to identify new SNPs for AF, further GWAS and replication studies must be done. For this reason, a GWAS was recently conducted in the Chinese population gene ID database, and 8 SNPs were associated with AF. Since GWAS relies heavily on large replication studies, and studies for AF have revealed 8 SNPs in the Chinese Han population, we applied Tachman assay to evaluate the association of those 8 SNPs with AF in a CCF cohort to determine whether these SNPs confer a risk of AF in subjects of European descent. And because many AF risk SNPs from GWAS conducted in European samples were successfully replicated in Chinese populations, we hypothesized that the 8 SNPs identified in Chinese populations would also confer risk of AF in subjects of European ancestry. genotype the DNA samples, TACMAN gene expression assay was used. TACMAN assays consist of a pair of unlabeled PCR primers and a TACMAN probe with two dyes, the FAM and VIC dyes. Each dye has different fluorescent signals and will bind only to one of the alleles. Therefore, both will bind to the heterozygous genotype, and only one will bind to the homozygous genotypes. This fluorescent signal is then measured by the ABI 7900HT machine running ABI sequence detection system. Running TACMAN assay, however, requires the PCR to be run first to amplify the segment of the DNA which we want to be expressed by the TACMAN assay. This will cause the signal captured by the machine to be more strongly expressed, and therefore the fluorescence of the fluorescent signal shown by the dyes will be much stronger. The PCR reaction consists of four main components: the forward or reverse primers, the DNA template, H2O, and DNA polymerase, with the reaction taking place in three main steps: denaturing at about 95 degrees Celsius, annealing at about 60 degrees Celsius, and elongation at about 72 degrees Celsius. These three steps are repeated 40 cycles to amplify the segment exponentially. After gathering the data from the TACMAN assay, we use several different tests to measure the randomness of the sample. The association of AF to alleles and the association of AF to the genotype using the Hardy-Weinberg equilibrium or HWE test, the allele association test and 2x2 chi-square test, and three genotypic analysis models: additive, common dominant, and common recessive models. These are two examples of TACMAN genotyping cluster plots. Each cluster represents a genotype, red and blue representing homozygous samples, and green representing heterozygous samples. The black X's represent undetermined samples that may have failed during PCR or were otherwise unable to be categorized accordingly. By comparing the percent occurrence of each homozygous cluster, we determined which sample had dominant and recessive alleles. This slide shows a summary of the four SNPs running the HWE test. As shown on the slide, RS3804987 displayed a high missing rate, which lowers the reliability of the sample. However, it still displayed a significant p-value of greater than 0.05, indicating that the samples were randomly mated. It is important to note that the alleles column is ordered such that A represents the minor allele and B represents the major allele. This table displays the results of the allele association test. The p-value suggests that RS2200733 has statistically significant association with AF. This table shows the result of genotypic association of the four SNPs using the three tests: additive, common dominant, and common recessive. A significant p-value is smaller than 0.05, and of the four SNPs studied, RS2200733 once again showed statistically significant association to AF, with p-values of 3.1 times 10 to the negative 9, 1.11 times 10 to the negative 6, and 1.6 times 10 to the negative 7. All values considerably lower than 0.05. We found that the SNP RS2200733 was statistically significant with AF in CCF-AF cohort with an OR of 1.85. This positive association was consistent with the published DBOS. The other three SNPs did not demonstrate statistical evidence for association with AF. Their effects were relatively low, indicating that larger sample sizes are needed from the results. And in the future, we will further analyze three negative associations in larger samples to confirm such associations by applying stronger OR values. We will also analyze more SNPs with suggested significant levels. And identification of new AF risk variants is a key to understanding the genetic basis of AF. So that will also be continued. Here are my references. One reference that was particularly insightful was common variation in atrial fibrillation, navigating the path from genetic association to mechanism. And I'd like to thank Dr. Fan Wang, Dr. Xing Wang, Dr. Xian Chen, and other members of Dr. Xing Wang's lab for their continued support and guidance throughout this project. And I would also like to thank the Starling and the Office of Government and Community Relations Civic Education Department for giving me this opportunity to be part of this internship.